I'd like to introduce our first speaker, Doreen Ware. I've had the pleasure of working with Doreen for over a decade now, and I can tell you that she is the person to go to if you want to understand about how a genome is sequenced, put together, and the insights that we can draw from it. She is a card-carrying genomic biologist. Under her belt, she has the assembled sequences and genomes of a number of plant species, but most importantly, the current reference genome of maize or corn, the most important agricultural crop in the world. So I think you're going to find that Doreen's the perfect person to start off this symposium because she has credentials both in agriculture and basic research, because she is in fact a scientist at the United States Department of Agriculture, Agricultural Research Service, but also a scientist here at the basic research Cold Spring Harbor Laboratory. So with that said, I'd like to introduce you to Doreen, who's going to take you on a fascinating tour of agricultural genomes.
Hi everyone, I'm going to share my screen right now and hopefully bring up my slides, which I closed. See, I closed them before we started, so give me a second here. So you should all be able to see this now. So I want to thank Dave for the introduction. As he mentioned, I'm from the United States Department of Agriculture. I'm from the research arm, the Agricultural Research Service. And I happen to be, and I'm very fortunate to be stationed at Cold Spring Harbor Laboratory. So what I'm going to be talking to you during this talk is I've given you an outline here. The first part of it, I'm going to be talking to you about agricultural drivers. I'm going to be telling you a little bit about, or a lot about, the last 16 years of working on maize. And then I'm going to transition and talk to you about some of the more recent work I've been doing in sorghum. And then I want to leave you in the last 5-10 minutes and talk to you about biology and big data, what I think about the future looks like. Because as we were talking about workforce development and the students and what we're going to be needing, things are very much shifting up, and the opportunity to participate in agriculture comes from many, many different fields. So some of the drivers for agriculture we think about is, with the growing population, we are going to need to increase agriculture output by 75% in the next 30 years. I mean, this is a huge, huge ask. And not only is it a huge ask from just developing and getting that increase in output, we're also having to do it under restrictions in the amount of resources that we need, as well as the changing climate. This means that we're going to need to think about how to prepare plants for new environments, how to think about maybe what plants we might be changing, maybe even domesticating new plants. We're going to need to think about preparing plants for diseases. I mean, if you look at what we're challenged right now with COVID, this is the same thing that plants are seeing as well, as we see new disease pressures. We also really need to consider, with climate change, how to collect and preserve the natural diversity. So maize is the highest worldwide production crop. Many of you are probably familiar that that's true with the US, but some of you may not be aware that that's also true for China. And the reference genomes are truly foundational tools for understanding what's happening for those plants and ensuring our long-term food security, as well as environmental sustainability. And I'm showing you this slide here because I want you to understand that the work that I'm going to be discussing with you has to do with a lot of people. Research isn't done in isolation. It's done very much collaboratively with many different groups who have very diverse backgrounds and expertise. And along my journey of the last 15, 16 years in maize, I really want to acknowledge my collaborator in USDA, Ed Buckler. And for the sorghum work I'm going to be talking about, I want to acknowledge my collaborator, Zhang Guozhen. So the maize genome is over 12 years old. I can't believe it. My son is a little bit older than that. And I think about him as being birthed at the same time the genome was. Now, when this genome was done, we were working on a very specific genome called B73. And the genome, when this was being sequenced, was sequenced using a back-based approach. And when we finalized the genome, half of the genome were in pieces of larger than 41 kb, and the other half were in pieces smaller than 41 kb. And we were able to stitch those pieces together based on physical maps that you'll see outlined in the ring in A. And the real sweet spot for us with regard to putting this together was that the average size of a gene is between about 5 and 6 kb. So when we're looking at pieces that were 41 kb in size, usually the whole gene was in there. And we could use these backs that we cloned and existing fingerprint contig maps from that and genetic maps then to scaffold this genome. So this first genome that we had was very incomplete relative to the quality that we're looking at today. But from that, we could learn so many things. So when we look at this genome here and we zoom in on it, what you'll see in green is the genes. And if you look at that green ring on F, that is the gene density. And what you'll see is the overall architecture of the chromosomes. You see that genes are usually towards the end of the chromosomes. And the section in brown there, which is ring E, represents the transposable elements that we saw, or the repeats. And we could see that there's usually an inverse relationship between the repeats and the genes. Where the genes are more dense, the repeats are less dense. The line B in blue represents recombination. And you'll see that there are different portions, usually the ends of the chromosomes have recombination rates. It's known that genes are hotspots for recombination. And the center of the chromosomes, the centromere is represented, the approximate position, by the red on each one of these, on each one of the inlays in A. It turns out that those low recombination regions are not easily exchanged out the genes. So it turns out that when breeders are working with things are in the low recombination regions are kind of tough to work with. If you look at this map too towards the center, we have information that overlays the genome of rice, which is smaller, about a 400 kb, and I think sorghum is at about 800, whereas the maize genome is 2,500, about 2,400 megabases, the size of human. Now in these grasses, it's known that the gene order and places are very similar between these genomes. And so if you look at G and H there, what you'll see highlighted or blocks are where the corn, the rice, and the sorghum genes are in similar positions. And this has been known for a while. And it's also how in the past, even with just genetic markers and not having the sequence information, people were able to transfer information between species by the anchoring of these genetic markers and knowing that similar genes were in these regions here. Now, we were very flattered that when we came out with our maize genomes, Jurassic Park also thought it would look pretty good with the Triceratops. Really, from a phylogenetic point of view, they probably should have picked something a little bit closer like a bird, but I like the maize genome there. Now, when we look at these sequences of genomes, by looking across the different genomes and within a species, we can see all the different variations that may exist in nature. And these variations kind of fall into three classes here. It's the letters of the single nucleotide polymorphisms, or SNPs. There are variations especially in plants, of the gene content, or what I like to think about the parts list in the organism. And it's known that there are many copy number variations where these genes are duplicated, or even presence and absence are sometimes called PAVs. Now, transposable elements are also very, very diverse in these plant species. And in fact, they were studied by Barbara McClintock and known as the jumping genes. And she received her Nobel Prize in 1983 for her work on transposable elements. And we now understand that these transposable elements actually play a very important role in the fluidity of these genomes, in moving genes around, as well in the regulatory sequences and impact the expression of genes. So maize is actually a tale of two genomes. So it turns out that many plants are polyploid. And in fact, maize had two genomes that came together. The polyploid can either be a duplication of the same genome and then those rearranged, but in this case, it was actually a fusion of two different genomes. And they came together, there was some rearrangement, some gene loss, and then it came back to being a diploid again. And if we look at this, we can use that information on an ancestral state. And actually, rice and sorghum are good ancestral states to be able to use as proxies for maize. And what we can actually do is overlay that, and we can identify which portions of the genome may have come from one of the ancestors. And it turns out that one of those genomes, in this case, it's the red genome, we could call the strong or the dominant genome, and the blue one potentially the weak genome, in that the weak genome lost more of its genome than the red genome. And in fact, even now, we see those signatures in the regions of, when we look at maize, that these regions are continuing to lose potentially from that initial ancestral genomes. Now, when the genomes came together and then they lost things, it turns out where the two genomes are similar. This is where the gray bars are. So now I have overlaid, what you're seeing here is these gray bars represent those duplicated regions. And most of these, you can see examples here where this is larger, this would have been the A genome, and this potentially the B genome. Now, when genes were retained in both of these copies, it can tell us something about perhaps how the genome is adapting. And it turns out that in maize, it retained copies of transcription factors, protein kinases, chromatin modifiers. And this is really interesting because this genome was pretty smart. What it actually did is it retained copies, or copies were being of genes that regulate the expression of other genes, of proteins that modify and regulate the function of other proteins, and proteins that modify the state of DNA, chromatin. And so if you think about it, when it retained copies of things, it kept two copies of things that regulated other things. So you kind of think of them as like middle managers, where they could regulate a bunch of genes. So what you have is the maize genome after this event had a whole bunch of copies of other genes that it could actually kind of flex around and play with and potentially use for adaptive responses. So humans have limited molecular diversity. Now, these were studies that came out from 2001, and so these are older data, but they still hold true. And at that time, it was looked at between human and chimp, there was about 1.3%. Well, between that same time, studies in maize suggested that the diversity between two maize lines is about 1.4. And in fact, if you think about it, then it means that each of these maize lines have more diversity than between human and chimp. And when we look at this, we already know that it's the SNPs or the genes, SNPs and gene differences. And we know in many cases that plants will locally duplicate these genes. And I'm going to show you an example from my colleague, Leon Cochin, who was at Cornell at the time he did this work, where an additional copy of a transporter will actually give those plants on the left the ability to grow on acid soil compared to the plants on the right. So the plants are very fluid. They will duplicate a gene, and if they retain all the regulatory sequences associated with it, they can use that to increase an expression. It's a quick way to basically change how much you have. When you duplicate a gene, you can also tweak and perhaps tinker on where and when it's expressed as well. So these extra copies give these genomes more opportunity to be flexible about changing where and when they might be expressed. So what we found at the time where we could use short read sequencing, and we had very skimmed data, we were able to develop haplotype maps of the existing maize lines and compare them to our B73 line. And we were able to call directly single nucleotide variations, but we were also able to look at structural variations using the short read sequencing by just asking the question, if one line had more coverage of an area, it suggested that it acted as a proxy of a structural variation. We would have called it a read depth variation. And at the time, back in 2012, we were able to show that these structural variations were highly associated with phenotype variation, and that if you looked at structural variations that were associated with the non-coding region, they were even more enriched for phenotypic variation. So we know that these structural variations in the regulatory regions are having impact in for phenotypes as well as in the gene regions. And we also recognize that having one reference assembly wasn't enough, and that we needed to really have multiple reference genomes to really represent the gene set and the diversity that existed within the maize population. So way back when, in 2002, I was working with the maize diversity group, and you can see how much I've aged since then, but we still work together. And not only is it the genome sequence, but more importantly, it's the populations that are generated that are able to help us dissect and look at genes and map genes that are associated with the different phenotypes. And this was a group that was located over different parts of the country and have been working together to develop out these populations. Well, there's one population called the maize nested association population, which was generated using 26 of these lines that were crossed to that B73 line that you saw. We had the reference genome sequence. And these 26 lines at the time, using microsatellite markers in 2002, were predicted to probably hold about 95% of the standing variation that we had in maize. And my colleagues did a lot of work to develop out these populations. And what they made was recombinant inbred lines that basically you're able to see now recent recombinations using these crosses. And each one of these families had about 200 individuals for about 5,000 rills, recombinant inbred lines. These populations have been distributed to Europe, to Asia, and the Americas. And many groups are operating on these populations and working on specific traits. So around 2018, 2019, we started looking at the maize NAM of could we then sequence these genomes? And this was funded by another NSF project, and we worked with collaborators at Corteva Science, previously at DuPont Pioneer. And we were able to then start working on the assemblies. And what I want to show you, I didn't mention, is in 2017, we published another version of the maize genome that was done with PacBio, these single molecule long reads technologies. And this genome had an N50. This is where half the genome, the shortest contig length, needs to cover 50% of the genome, or half the genome is covered with contigs larger than this, of 1 megabase. Now, we were so excited because we were part of the 1 megabase club, which Human was part of, with having an assembly. So now our assembly contiguity went from 41 KB to 1 megabase. We were so excited. But our new genome that was just released this last year is now at 50. So you can see how the technologies has changed. And these technologies have changed because of the chemistry and also engineering associated with the sequencers. so that it's now the average length that you can get off these machines are in the 20 kbs for the assemblies. And we know that these retrotransposons are in the 15 kb. So once we were able to break that barrier of the 15 to 16 kb length, we could actually assemble all of the retrotransposons. So I'm not going to tell you any more about the retrotransposons, but I want to talk about the genes. So in these 26 accessions, we've been working on the annotations, and we feel that we have about 100,000 genes in these 26 assemblies. And my colleague Candy Hurst at the University of Minnesota, she has been working on the pangenome complement of this. And what Candy group has looked at is much like that red genome and the blue genome that I talked about earlier. She's called subgenome 1 and subgenome 2. And then genes can be syntenic or not syntenic. And what you see here is of those 100,000 genes, about a certain percentage of these are in all 26 genomes, and a certain number of them are in just one genome. And this starts to go into the concept of having a core gene set, some dispensable genes, and private genes within the population. Although they use the name dispensable genes, I really like to think about these genes as being the adaptive genes. And what we're able to see is that across these 26 lines, we have about 27%, almost close to 30% are core genes, and the other ones are segregating in the population. Now, when we look at these genes in the context of evolution, if you look up here in pink, these are the gene family, the genes that are in gene families that are relatively older, that are in all plants. And genes in the blue-purple color are genes that are in the Poaceae and are part of the monocots here. These genes in yellow are genes that are associated with genes that will be in families between sorghum and maize. And then the ones you see in green are genes that look like they seem to be in families that are explicit to maize itself. And what you can see then, you can think about this, is as these new genes are evolving, these new genes, and they're being selected for, they're sort of like a birth and death rate associated with them. And the ones that are going maintained and being constrained and selected for are likely to have some fitness impact or adaptive response and rewiring that's happening. So I like to think about, when I look at the genome, it contains a life history of the species. And when I'm looking, what you're looking at here is traversing of what's happened in the domestication of maize, what the human pressures have been to go from this ear size with a few grains and that had very hard coatings to the current version that we have of an ear in maize, as well as a plant that has tillering and a bit weedy and branched, to a plant that's very upright and has erect leaves that allows you to plant a very high density. You can look, depending upon the genomes that you're looking at, you can look and see signatures of what fitness happened potentially during domestication and then happened during improvement here. So at this point in time, I'm going to switch over and I'm going to start talking to you about how we can potentially use the sequencing, the technologies, and start to build what I think some of the biology and the biology-enabled agriculture. So when I talk to growers or I talk to the commodity groups, I work with several commodity groups. I work with maize, sorghum, rice, and grapevine. When I talk to the growers, you know, the big thing is yield. I want yield, I want yield, I want yield. There's one trait: yield, 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 yield. If you talk to somebody like grapevine, depending upon whether it's grape for wine or grape for table or grape for juice, it's going to be more of a quality trait. In fact, the grapevine community involved in wine, it isn't about yield. It really is about quality. And so these traits are very high level, very complex, after the plant has grown and seen multiple environments. And really what we're really looking at trying to do is be able to do that old-fashioned genotype to an environment, and the plant doesn't care if its climate or management practice on the farm will give you a phenotype. And we want to be able to go in both directions. So I'm switching to work that's been done with sorghum with my collaborator, Jean Grosjean in Lubbock, Texas, where he created an EMS population in sorghum. And what this introduces random single nucleotide mutations. And this technology has been used since the 30s to introduce new novel genetic variation into agricultural species. And then these genetic variations have been moved forward into the breeding programs. And so this is not considered a GMO approach and has been used for many years. Both Zhenguo and I shared a postdoc who went between Cold Spring Harbor and Lubbock and spent some time with us, and now has her own lab in Texas Tech. And she was involved in sequencing these pools. And from these sequencing pools, we could identify new variation that was introduced. And the important part is at the bottom of the slide, where we have 22% of the mutations we identified with very high confidence is in 95% of the genes. And most of half of the genes, more than half of the genes, have potentially 2 disruptive mutations. So I'm going to tell you some of the stories that we've been doing to try to dissect some of the complex traits like yield down into smaller component traits. And this one has to do with fertility or potentially flower architecture. Now, when Zheng Guo was walking the fields, what he actually saw was a phenotype called multiple seed. And in multiple seed, what happens is that the normally in the sorghum plants, the sessile spikelet is fertile, but the pedicel spikelets that are tending are not. And in this plant, all of them were fertile. So in looking at this, we were able to make new populations, do sequencing of those populations, and based on the segregation, we actually identified the gene that was involved in MSD1 as a transcription factor, TCP, a TCP transcription factor. Well, the TCP transcription factor is actually an old friend. It turns out that the TCP transcription factor was identified as one of the transcription factor families involved in teosinte branch that John Dolby published in 1995. And so if I would really highly recommend, if any of you are interested, to Google Secrets of Plant Genomes Revealed, and you should come up to an NSF site, and they have some fabulous videos on the dynamic corn genome, of which highlights the work that John Dolby has done and the selection of a few genes about that are associated with that transition and domestication. But we had 2 more alleles. And it turns out that those 2 more alleles, we did the same thing. We were able to make populations and then sequence from these, and we could narrow down to 2 different genes. They were both biosynthetic genes that were involved in a plant hormone pathway, jasmonate. And it turns out that what happens is the jasmonate, the plant hormone, will actually, and where and when that's and how much is expressed, will actually make a flower, the sorghum plant, have the infertile flowers. And in fact, we could apply this hormone to the mutants and revert them back to the having only the sessile spikelet being fertile. And in fact, Nick Gladman in the group was able to demonstrate that this transcription factor binds the promoter of these 2 genes as well as other genes. So I want to move on next and basically tell you and talk about other components of yield. So having not enough nitrogen will actually impact the yield here, but actually having too much nitrogen is really bad. Right? And we can see this as examples of agricultural runoff. So one of the things that we wanted to understand is what are some of the genes that might be involved in nitrogen and may impact fitness for nitrogen. And the approach that we actually took with our collaborators at UC Davis was to build an Arabidopsis regulatory network. And we did that by basically using information of things that we knew were involved with nitrogen, like the nitrogen transporters, genes involved in metabolism, and a few other genes. And we were able to use an approach called a yeast 1 hybrid approach, where we could clone the promoters of these and then screen them with the transcription factor library and identify the transcription factors that would bind to the promoters, developing this network. Then Andrew and Christos in my group were able to integrate expression data and build some and expression data for treatments and where and when in a plant that these were expressed, and they were able to develop predictive models that would suggest which of these transcription factors may have impact. And then doing that, we were able to then leverage existing Arabidopsis loss of function mutants from tDNA populations, order those transcription factors. And Ali in Shaban's lab was then able to test to see whether they had a phenotype. And we had a very high predictive outcome of being able to show which genes were having impacts on performance in limiting nitrogen for their root system architecture, as well as their biomass and flowering time. And based on that, we were identifying some key transcription factors that may impact nitrogen fitness in the plants. And it turns out that some of these are actually progressing forward in some crops. So with changing climate and increasing temperature also impacts yield. And this is a map of the drought monitor from 2000 to 2010 to 2015 to 2021 in early January. And what you see is a trend of increasing temperature and drought. So what we're seeing is a change in the overall climate that's going to be basically impacting what plants can grow and the need to adapt these. And one of the ways that we can use is a reverse genetics approach, where in Arabidopsis, we knew genes that were involved in epicutical wax. We could look at those in our sequencing pools and say, did we have any genes that might have had change in their amino acids? And then look and screen these in the field and and screen them to see if they had any change in their wax. And in fact, it was predictive on having shifts there. We also had some other genes that were previously identified that allowed plants to be more heat tolerant, and we could show the loss of function of those actually had improved performance in the field. We could also go the other way around and look at plants that had different water use efficiencies in those mutant phenotypes. Much like we saw the multi-seed mutants, we saw plants that had different fitness impacts in the field, and we were able to use the same approach to identify, in the end, the same very similar groups of genes as that we found when we used the reverse genetics approach. So I'm going to shift to another example with climate change. We have impacts and disease pressures, and with yield losses, what farmers will do is they'll spray more pesticide. This has a huge direct cost, a large impact to the environment, and potential impact to human health. Now we know that disease resistance genes from many studies are rapidly evolving. I can't tell you the work we've done in rice and the time we have, but what we know is that they're good candidates for structural variation because these genes are usually seen in clusters. And so they were a really good target to use for these reference genomes. And in 2013, their sugarcane aphid almost devastated the sorghum community at that time. It wiped out many of the crops. And in fact, it was some of the hurricanes and things that they would believe brought a new population of aphids over from the Caribbean that was potentially could be responsible for this. My colleagues in USDA made populations with a line that was tolerant and a line and the reference assembly that is intolerant that was available at the time. And they were able to map this gene to the top of chromosome 6. In the last 2 years, we started then to work on this reference assembly with our collaborators at Corteva. This is the change that we're seeing. In our new reference assembly, we have whole chromosomes in a single contig. It is just amazing. This is work done at showing a gene neighborhood view. It's very complicated. This is a pan-genome site that we have up and started. The important part for you to notice is this thing boxed in the dotted in blue here. This is the insertion, and you can kind of think about this as a zoomed out view of a housing complex that are all the same here, where the genes are all the same up above here in these other lines, but here you see the insertion here. That insertion is represented here, and when we align resequencing data from multiple other accessions, very wild varieties down here, land races, and improved lines. What you see here at the very top is the 2783 with its own resequencing data alignments. You see all the genes are there, but you see these blue things that represent where there's no coverage of these genes, showing the potential structural variation. But there are a few lines in there, accessions in here, that may also have resistance, and we may also be able to use to dissect which of those genes are associated with it. So as we start to move forward and we look at exploring genomes with an eye towards breeding, what I've hoped you've seen is that we can use approaches like association analysis that was used with TBE1. It gets genes of major effects, but to get to some of these genes that have minor contributions, we're going to need to use regulatory and inference networks to be able to drill down to them. And we have the power to use as well as comparative analysis. And our real target is understanding the biological mechanisms in the systems level to be able to improve the breeding models and support for genetic engineering in the future. You know, over the last 16 years, what we've seen is this amazing drop in cost of sequencing and the length of the sequencing reads. But with that has come this increasing need for data management and use of computes. So while we've seen the reference genome drop from $50 million to $2,000 to $30,000 right now, using this technology, we've had an increasing need to use computational data management approaches. And this is where biology has really transitioned to an information science. And this was an outcome of a keystone meeting that I organized with Lincoln Stein and Michael Schatz. And what this is really highlighting is we are streaming data from all different levels, all the way from the single cell level to interactions between organisms, to weather, spatial, satellite data, and whole field data. And we can basically stream genomes, stream environmental data now. And we need the systems in place to support managing that data. We need ways of basically storing and standards to allow us to access this, to actually leverage machine learning and modeling and be able to deliver that to the domain scientist. Now, in November, something really exciting happened at the recent CASP assessment, where they have this basically they basically test every year the algorithms for protein structure predictions. AlphaFold was introduced. And this is the quote from the former chair and founder of Genentech. AlphaFold is it's in a generation advance, predicting protein structures with incredible speed and precision. And this leap forward demonstrates how computational methods are poised to transform research in biology and hold much promise for accelerating the drug discovery process. Now, I want you to rethink how we might think about it. I've told you about disease. This is really the impact. This is from a 2011 economic assessment of just the amount and the cost associated with grapes alone in California to the industry. Not only that, think about all the pesticide that's going on this, right? And what that means for our environment, our health. Well, it turns out wild grapes have natural resistance to genes, okay? But with the domestication process, they have shed some of their baggage. Some of their baggage is they taste really, really bad. Okay, they also are no longer, the domesticated one is a hermaphrodite, so you can't sex these early and they don't mature until 4 or 5, 6 years old. Okay, so there are some issues then associated with this. For disease resistance genes, it's a concept of a gene for gene interaction, where there is a pathogen that elicitor and then a matching gene within the plant that recognizes it and sets off the immune system. Could we even begin to imagine a world where we could model, much like drug discovery, the protein ligand for the models for these disease resistance genes against their pathogens, their fungal pathogens, their bacterial pathogens? And then we can imagine a world where we can be doing this modeling and then applying it in a way that we could stack these using some of the technologies that we have. So the next green revolution will be data and design driven. And I think the data now exceeds our human capacity to really formulate these hypotheses. And we need to think about this. We really have a responsibility to use some of these new approaches to help inform crop breeding, come up with different fermentation systems, and pest and disease management. And the way that I think about this is we will be adapting what nature has already learned about, that life history that I talked about, and that what we will be responsible for doing is designing the space that nature missed. Now, this is a slide courtesy of my colleague, Ed Buckler in Cornell. And it shows that basically what we've done for the first, many years here as humans were evolving is we've been selecting plants that we've been looking at. More recently, we've used quantitative genetics to apply and accelerate that. And more recently, we've been using molecular approaches of marker-assisted breeding and whole genome selection is used in cattle, several other animals, and several other plants. But we have the possibility of designing these genomes based on the information we know and the interactions between the microbes and the agricultural species. So I just want to leave us with that while CRISPR has the power to enhance breeding by rapidly customizing and optimizing crop productivity, genome biology provides the insights to drive that translation. And this is the thing that really excites me. And, I think this is just an exciting time for students and things who are entering the workforce about all the potentials that they have in the future to contribute. So I'm talking to you about work that everybody else does, and this is basically to acknowledge my group. I have an amazing group of staff at the lab who have been working on the genome assembly work, genome annotation, which I haven't been able to talk to you about, infrastructure and standards that are needed to support stewardship of these genomes, as well as the group that's working on functional genomics. And I also have to acknowledge my colleagues longstanding at Cold Spring Harbor, specifically Dick McCombie, who I've worked with very many years, who've helped me on the genome work, as well as Rob, who you'll be hearing with Dave, Tom, and Jim. And Dave has been an excellent colleague for the outreach, education, and learning. And we wouldn't be able to do this without the farm and as well as the HPC resources. So I always joke in my lab, I have 2 farms, the compute farm and the real farm. All right, so I'm going to leave it here and I can take questions.
Hi, Doreen, I'm going to help you now so you don't have to fidget around too much. We had an early question that was submitted, which is, do scientists understand what determines a weak versus a strong genome when you talked about recombination?
So when I use the term a weak or a strong genome, someone actually, when I first wrote that, they called it the bully genome versus the bully genome. And so this was a genome that's basically taking over. It's suppressing the other genome. So there are some things that are suggesting what's involved with that, and I believe my colleague Rob Martinson, who's going to be talking about this, may actually understand some of the keys to that. But some of the suggestions may have to do with the repeats, how things are basically the repeats in the genome and which repeats are being selected for, whether it came from the maternal genome or whether it was paternal genome, and what happens in the nucleus at the time. So there are some hints for that right now, and I think that Rob may tell you more about some of those things.
Here's another question, totally different. Why is it that CRISPR modification is not considered genetic modification, and that plants that are modified with CRISPR should be labeled GMO?
So it depends which country and where you're at on the policy of whether a CRISPR is being defined as a GMO or not. So in the US, a definition of CRISPR being used depends also on the amount of sequence that is being put into the plant, the size. It's also potentially dependent upon whether you could have bred it normally. It may have taken a long time to breed it, but whether you could have introduced it through traditional sexual practices, or whether that variation would have been seen in nature, it makes it whether CRISPR or not would be considered GMO. So let's just talk about grapes. I'll give you an example. In grapes, grapes haven't really, the grapes that are used for wine haven't really bred for hundreds of years, right? The joke is they haven't had sex for 500 years, right? But they've had a lot of damage because they're clonally propagated. What if you could make Chardonnay look like the Chardonnay of 500 years ago by just changing one or two nucleotides in it, because those were shifted due to mutations that happened over time, right? So this is where these things happened in nature previously, and we're just reintroducing them. I'm not sure if that helped with the answer.
There's a different question that came up, which is, how do you do gene identification using RNA-seq data?
So RNA-seq data is monitoring the transcripts that are coming from the genome, right? So those transcripts can be mapped back to the genome, and we can use that in mapping it back to the genome. But the transcripts themselves can really be a proxy for the genome as well, because it's a downstream product that's spliced from it. So I would say that the RNA can be used as a proxy of a small portion of the genome that represents the transcript.
Doreen, I'd like to highlight one of the concepts that you brought about in your talk. Many times biologists think that the variation that we do in breeding plants or improving crops or animals arises by mutation, because we have this classic idea that mutations happen and some of them are null and some of them are good and some of them mean nothing. But you brought up this idea of the sort of genetic history or natural history of a plant. And there's a concept called standing variation, which is very important in plant breeding. So maybe you could just elaborate on how plant breeders use what is called standing variation.
So one of the reasons why we wanted to sequence as many of these genomes that we could, and we identified in maize that there are 1,000 genes in there. So if we had just looked at that maize genome, that B73 genome that we had that had about 39,000, 40,000 genes in it, you would think, oh, there's only 40,000 genes in the species, in the population. But then when we looked at the 26, you can see that there's a lot more variation in the individuals than you saw in that one, that would be the portion of standing variation that we use. And so when the breeders are looking at this, they have a certain amount of variation that they already have in their existing population. And a lot of times what they will go out and they will look at wild varieties and wild relatives, because they may have some other genes and traits that are not in their standing population, and they'll go and they'll grab that and they'll bring them in. And this is where they can introduce new variation that they don't have in their variation that exists in the populations that they're working with. Is that what you were looking for?
Yeah, that really, in many cases, plant breeders are using existing variation that exists among individuals rather than, new mutations that have occurred recently. These are ancient variations.
That's right.
We have a couple more minutes perhaps, but before we end, I'd like to remind everyone, if you would just look in your chat box and follow the link to take a 30-second survey about this talk that she's just given. And we'll have a different survey for each of the 3 talks. So we might have time for one more question if somebody would like to ask a different question.
So I see a question. I'm looking at I'm looking at some of the questions. How do our gene identifications? Oh, I'm sorry. It was our gene identifications from RNA-seq data. I'm sorry. I
misinterpreted. I thought it was a typo. Sorry.
Okay. So how do our gene identification from RNA-seq data? Well, actually, that's a good question. It turns out it's really hard to identify different disease resistance genes when you use RNA-seq data, because the RNA-seq data is broken up into small pieces, and then we have to put it back together. And when you look at a lot of these R genes that are these adaptive genes, it turns out that they're extremely similar. And in fact, one of the really beautiful things about the long-read sequencing technology we can apply to the transcriptomes as well is we don't have that issue of breaking the gene up and then putting it back together, and using a long-read sequencing technology to get full-length transcripts. We are able to distinguish the R genes in a way that we cannot do that when we use RNA-seq data.
Here's an interesting follow-up question to one that was asked before about gene editing. Can a gene-edited species crossbreed with natural species?
In theory, it can, right? So if a gene-edited species, so if you introduced a change in a nucleotide using a CRISPR technology that does not leave a footprint in the genome, it should be able to breed with an individual that hasn't been edited. So one of the concerns with editing, and I think that this might be what someone might be wondering, is that while editing is possible in species, there is concerns about editing that can happen in species, in crops, where there are wild and weedy relatives that are grown in the same area as the crop that you may edit. Because there is concern that if you edited a crop that has a wild weedy relative, like for instance, sorghum and Johnston's grass, and you made sorghum actually an absolute fitness performer with that edit, could that edit then, could that sorghum plant intercross with the wild relative and then actually make that wild relative that's weedy have a better performance and make it tougher to get rid of the weeds? That is possible. And so even though CRISPR is not regulated as far as a GMO, there are reviews and processes in place as you introduce new varieties to the market that consider what the impact is on what that potential impact of that crop may be. So even if something isn't GMO, and would be available from CRISPR, there would still be regulatory processes that would review that.